Hey guys, quick biochemistry basics here. Let's talk about enzyme kinetics. Enzyme kinetics is a study of how enzymes bind their substrate and convert them into a product. During enzymatic reaction, the enzyme binds the substrate and forms enzyme-substrate complex. The ES complex is usually in a transition state in which substrate is converted into a product and the product is finally released. Now here comes a very important term, kcat. The number of substrate molecules converted into product per active site of enzyme in 1 second is called turnover number or kcat. The units of kcat will be second inverse. The study of enzyme kinetics becomes easy when we try to understand the reaction by plotting a graph. So what all graphs can we plot? In our case, the enzyme is acting on a substrate and the substrate is getting converted into a product, and this occurs with respect to time. We can plot this phenomenon simply as concentration of substrate or products versus time. As the substrate is consumed, the concentration of substrate decreases over time, and as the product formation continues, the concentration of products increases over time. If we take the slope of the graph, then what we get is the rate of reaction. The rate of reaction is the change in concentration of substrate or product per unit time. As the substrate concentration is decreasing, we use negative sign, while in case of products, we use positive sign. The unit of rate of reaction will be concentration per unit time. We can also call this as velocity of the reaction as the concentration is changing with respect to time. Now it turns out that the velocity of reaction changes when substrate concentration changes. This is something that is seen experimentally. With an increase in substrate concentration, the velocity of the reaction increases until it reaches a plateau. This is called maximum velocity or Vmax for that particular enzyme. Now let's divide Vmax by half and find the substrate concentration at which this is occurring. The substrate concentration at which half of Vmax is achieved is called Km. Remember, I again repeat, because lots of students make mistake when they're asked the definition of Km. Km is the substrate concentration at which half of Vmax is achieved. The ratio of kcat by Km represents efficiency of the enzyme. This ratio is called specificity constant. Higher the ratio, the enzyme will be very efficient.